Hello and good evening, everyone. Can you hear me? How are you all doing? Please let me know quickly in the chat if you can hear me, if I'm visible, if I'm audible, if your energy is high. So, this is the midweek. I hope you're having a good Wednesday, a good week. So, are you all eager to learn? So, quickly let me know, guys, please, in the chat if you can hear me, if I'm visible as well. Good evening, Preeti. Good evening, Anushka. Thank you so much for the confirmation. So, are you all ready, career ready for bioinformatics? I know we've talked so much about it. It's a never ending field, it looks like. Always we have new things coming up. So, are you all very eager to learn? Are you all ready to become job ready, career ready in bioinformatics? Okay, so we'll be discussing the must have tools and skill sets for the modern computational biologist. So I hope you all are really ready for this session. It's going to be really amazing. We have our in-house experts with us who kindly joined us today to share their wealth of knowledge. Okay, so if you're ready, let's begin. So bioinformatics has rapidly evolved from a niche expertise into the backbone of modern biological research. So today it is a core requirement for biologists and researchers driving breakthroughs in pharma, genomics, and personalized medicine. So if you watch Sir's videos regularly, you would have heard these terms, genomics, personalized medicine. So right now we are in it, okay? We are in the thick of it. So with the rising integration of data-driven science, the demand for computational biologists is surging. And by 2030, that is less than 5 years from today, over 70% of biology-driven research jobs are expected to require computational literacy. I think you would have observed the same. So if you see all the jobs that we post on our Biotechnica portal on a daily basis, there are so many institutes and companies always looking for bioinformatics skills, computational biology skills, AI, ML. I hope you all have noticed it, right? So sudden uptick in this. So prior to 2020, not much talk of bioinformatics was happening. Post COVID, everything has changed, and now, like I just read this fact, that within the next 5 years, over 70% of biology driven research jobs require computational literacy. So now the decision that you all have taken to attend this webinar and learn all about this is the right decision. It shows how keen you all are and how serious you are in pursuing a career in bioinformatics. So like I said, we have two in-house speakers with us today. Our speakers are Dr. Elamathi. I'm sure you've heard of her. You've interacted with her. Hello, ma'am. Good evening. Thank you so much for joining us.
Good evening, Dilipshay. Good evening, everyone.
Okay, so a little bit about Dr. Elamathi. So Dr. Elamathi Natarajan is a passionate bioinformatician with expertise in computational biology, genomics, and data-driven research. She holds a PhD in bioinformatics from Dr. APJ Abdul Kalam Technical University and an MBA as well in information systems management. Okay, so she was an assistant professor and an HOD, and she has guided students in both research and academics. Now at Biotechnica, she has been instrumental in developing bioinformatics pipelines, applying machine learning to genomics data, and supporting advanced research projects. She's also a recipient of the ICMR Senior Research Fellowship, and she continues to advance innovation in bioinformatics and applied life sciences. Now, our next speaker that we have for today's session is Dr. Neeraj Kumar. I'm sure you know him as well. Hello,
Hi,
sir.
good
Good
evening,
evening.
everyone. Hi, good evening, good evening.
Yeah, thank you so much, sir, for joining us. So, a little bit about Dr. Neeraj. So, Dr. Neeraj is a computational biologist and bioinformatics specialist with a strong focus on AI driven drug discovery and structural bioinformatics. He earned his PhD from CSIR, IHBT, and ACSIR. I'm sure all these names you've heard of, and pursued postdoctoral research at Pennsylvania State University, where he developed computational algorithms for immunoglobulin analysis. His expertise spans machine learning and deep learning applications in drug discovery, cheminformatics, and molecular modeling, using tools such as TensorFlow, PyTorch, AlphaFold, Rosetta, and GROMACS. So he has publications in leading journals like Journal of Cheminformatics and Medicinal Research Reviews, and he brings deep insight into how AI and bioinformatics are reshaping the modern drug development. So these are our two esteemed speakers today. So we'll conduct the session with them. At the end, we'll have an Q&A as well. So you can drop in all your questions in the comments. And also stay tuned because we have the free guide. Okay, so we'll be dropping the link for it at the end. So stay tuned to get your free bioinformatics guide. So without any further ado, let's begin the session. I am handing over the controls to Dr. Elamathi.
Thank you, Dilakshi, for the introduction. Good evening, everyone. And thank you so much for all of you for joining the webinar today. Yeah. So today we will be discussing about one of the very, exciting session, I would say. Okay. Everybody, just a moment. Yeah, so let me share my screen. Is my slides are visible to everyone?
Yes, ma'am, the slides are visible.
Yeah, okay. So, as I said, today we are going to see one of the very excited session. Okay, everybody has a myth that computational biologist means only that you should develop a pipeline, you should know how to analyze the data, etc. But there are certain. This is not what the employers are actually looking for. There are some special traits like nowadays everybody expects from the computational biologist. Okay, so today I am here to help you out to tell you about tips and tricks how you can master a good computational biologist and you can develop your career development skills. Today we are going to discuss that exciting topic with you, all of you today. Okay, so I would like to share my own experiences and also like how I'm seeing many people's forefront years in front of me, like a freshers like you or intermediate professionals, experienced professionals in front of me, how they are growing up in their career, all their experiences and views. Today, I'm going to share with everyone today about that. So, actually, if you think we were living in the era, it's completely where sequencing and all other biology related wet lab experiments is generating a huge data. Okay, so nowadays biology is not only about the experiments, so there are a lot more other things about the data and data analysis. Now the computational has become one of the important core techniques too. So just give a moment of think that how much amount of sequencing data being generated in single day or worldwide. If you see, it is comparatively much larger. Okay, so even entire human genome project, how much it has been generated, actually it has been generated more than that too as well. Okay, in worldwide. So actually it is mind blowing, right? So how to analyze those data? Okay, so here is the challenge comes here. Okay, so when the data remains as data, it is a meaningless one. Okay, so we should use the right skills to interpret it, analyze it, and also connect and interpret more related to the data convert and transformation into a biological significance interpretation. Okay, so when data remains just data without analysis, it is actually just a noise. In our language we say it is just a noise only. Okay, it is not actual data. Okay, so now we all should become a modern computational biologist. Okay, their role comes in actually. Okay, so today thoroughly through this session we'll be exploring like we should not act as a pipeline operator, how to solve a real world problem Okay, how will you become a hybrid scientist? Okay, where you can able to bridge the biology and as well as computational part. Okay, so I would like to give you a clear essential skills which is actually required to build in your career right now to make your portfolio much better. Okay, shall we start? Yeah. Okay, so nowadays if you see, like people's have one new demand everybody had actually. Okay, if a biologist means, in the past we have a tendency, okay, that every wet lab scientist should be there, wet lab researchers should be there. Okay, so everyone has their own separate roles they have. Okay, so you should either pipette in the wet lab, okay, or else you should code in the dry lab. Okay, but that separation being broken down nowadays. Okay, so today employees are actually looking, actively searching for hybrid scientists they are looking for. The peoples who are really who understand the wet lab context. Okay, but they should also able to know how to analyze the data in the terminal, and how they can perform the alignment with the data, and critically who can able to interpret the output. Okay, so employers are really searching for such kind of hybrid biologists nowadays. Okay, so why this trend is going on? Okay, why? Because nowadays in all of the global projects worldwide, data is a main optimum thing. Okay, so with the data is a central part where every single results will give that valuable information in the research. Okay, so now you see, just one single sequencing data, a single sequencing run in the wet lab, they can able to generate terabytes of data. That is much interesting, right? So here no one particular single PI or bench scientist can handle this alone. Okay, so all the employers are seamlessly nowadays, they look for some person who can able to understand the biological concepts, who can able to handle those wet lab data and the computational tools in an effective way. Okay, you just think in this way. Okay, imagine that you're studying the cancer. Okay, now as a wet lab scientist, what you will do? Okay, so like they just observe a gene expression data. Okay, so they are gene expression change, how it is there. Okay, so if you do the wet lab experiment of gene expression, and if you will store in the those results in your computer, or if you are publishing that results into paper, it will just remind the results in paper only, but that won't turn into a actionable insights, right? So here come the computational biologist role will play a role. Okay, so here some person really he need to work into the data, he need to convert those rates into a actionable pathway, or he or she can able to find out the therapeutic targets. Okay, or else any kind of research hypothesis. Okay, so that not only remain in paper, actual discovery will come outside on a roll. Okay, so the demand is not just only for the coders or biologists. Okay, it is for all kind of integrator scientists, it is there. Okay, so such kind of people who actually can bring both the worlds together. Okay, that is none other than, it's all of you, okay, who is listening to today's webinar. All of you will be a future computational biologist. If you will definitely master in this core competencies, you will be one among them. Okay, so now I will for all of you to think about where you are standing exactly. Okay, you need to rate about yourself. Okay, then only you can develop your skills into next level, and then you can get into the world of job. Okay, as the world of job is in a competition, you can enter into that. Once you rate about yourself, you should know about yourself. Okay, so for all of you, I'm just giving one task. Okay, just rate about yourself. If you are a wet lab person, okay, please rate yourself in the rating to 1. Okay, and if you are a computational skills, how much you have, you please rate among yourself between the 10 as highest. Okay, so now in the between the wet lab and dry lab, where you are standing, you please rate yourself and improve yourself to the career world. Okay, so you should be the next one to be employed over somewhere, right, in big companies or academia or research industry, everywhere, right? So you should be a act smartly to be a hybrid biologist. Okay, yeah. So now we will discuss why these computational skills are actually matter. Okay, so we we address this big question: why can't we just rely only on pipelines? Okay, why we require some other additional computational skills are really matter? Okay, so we should understand the truth. Okay, so pipelines, it is a fantastic, uh, because all the steps are automated. Okay, so if you develop a pipeline, so you can be getting to hired somewhere as in a nice position and all, as a software engineer, everything. Okay, but here every steps will be automated in pipeline, like start in NGS data analysis, starting from alignment, quality control, variant calling, and completely the pipeline will be fully aligned and it will be automatic. But one bitter truth is that the biological significant result interpretation, it cannot explain what the result you got. Okay, how you are mapping it. Okay, what is is there any result bias is there, if any sequencing artifact is there, such kind of interpretation, or it has been done in a very quality way, the result has been done, that you cannot say directly. Okay, yeah. So let's take one example we'll take. Okay, I have seen one team, they were running on RNA sequence pipeline experiment they were doing. Okay, and after a while, proudly they were reporting that hundreds of differential expressed genes they got it. Okay, but actually when we look closer, they have turned out, their control and case samples, swapped between that. Okay, metadata mislabeling was being done by them. Okay, so the pipeline but they didn't catch it. For them it is just a data. Okay, so pipelines, they don't actually think how we human beings think. Okay, so here in this case, computational really matters a lot. Okay, so here in computational skills, you can question the results, what exactly you have got. Okay, and even you can identify errors what is there, and you can better interpret your outputs. Okay, and biologically how they are beneficial, you can connect them in a proper way. Okay, it is without the critical thinking, you are just pushing the buttons, in the with the software or tools, it doesn't matter. Okay, so employers actually know this. Okay, that's why they look for real computational biologists, not like a person who just know how to use the tools and softwares. Okay, so here the computational biologist here, they should know whether the data makes sense biologically or not. Okay, so are the patterns are consistent with the known mechanism, or any other new novel mechanism is there, or you should also do some additional checks, validation, cross validation, what you have done, what the discovery you have been getting to is the real thing, or any kind of biased results you have got into. Such things, the clarity you should have on the things. Okay, that's why the computational skills matters a lot. Okay, so here the proper mindset of the shift also we need. Not only the pipeline maker are required in the skill, so computational skills who understand with the proper context, that such kind of persons is really needed. So if you are wanted to get into good job with good things, so you should not work mechanically learning the things. Okay, you should understand the context and apply where it is needed, when it is needed, and how should be related to the biological results. Okay, this matters a lot. Okay, yeah. So next thing is very basic thing, or good quality, I would say, you should master the biological databases. The basics are very much needed, even though now groundbreaking research are going on. The main the basic science remains the same. Likewise, in bioinformatics, the basic science remains the same, with mastering the biological databases is one such quality. Okay, so the first major competency, like every computational biologist should have this, mastering all the biological major databases. Okay, for every application we have a specialized databases we have, so you should master that one. Okay, so you should think that databases are will act as a library for our biology. Okay, or these repositories will really beneficial in most of our real-time life research. It is very much needed actually. See, in every research, data is more important thing, right? These databases will provide and all those necessary information which is research. Okay, so but unlike a normal library, here usually library, what you will find, books in the shelves, right? Here exactly we find in biological databases a dynamic, interconnected, and constantly updated new informations of data you will loaded in the biological databases you will get into. Okay, so some of the key databases which actually mainly we use for primary research is NCBI. Okay, so here in NCBI, this is an entry point of finding the sequences. Okay, either gene sequence, protein sequence, nucleotide sequence. Okay, and PubMed. PubMed is one thing where you will get articles. Okay, complete literature survey you can get over there here for healthcare related articles, and also other technology related articles you will be finding over here. Next is SRA database. In NCBI you will have genome related, and also the hotspot of NGS data will be available over here. Next come is your UniProt. Okay, so here it's go to for protein function domains, and also pro-structure modification information exactly will get here. Next is Ensemble database. It's actually a genome browser which helps you to to go through, explore annotation, orthologs, and also any kind of variation related information you will find here. Okay, so only these key databases are not enough. Okay, to know the existing thing, you should learn, query them efficiently, you should know. Okay, so when you look up a real gene, okay, you need to check which transcript is the reference in the database. Okay, and are there isoforms, are they related with any association, that you should know. Not only these key databases, we have some more specified databases we have, you should gone through specifically over that. Okay, here we cannot exactly tell how many time projects, many of the cases I have seen myself where projects have simply gone wrong. Okay, someone has chosen wrong transcript ID, or some of the peoples have ignored the assembly variations. Okay, such small informations, that has been ignored. Okay, so some one mismatched ID, that could spoil the whole detail, whatever has been done with the work. Okay, so spend time in mastering database is a smart decision, and also make it a habit. Okay, whenever you read a paper, you can see the gene of interest, go to the database, search it in a nice way. Okay, look it up, at least some 2 to 3 databases to cross check the informations are right, then you can take them to the research. Okay, yeah, apart from key databases, there are many more databases are yet to learn, master all those things. Okay, if you see, NCBI will remain as a mastery of everything. To learn a NCBI, a person should really spend 1 month completely to learn about complete resources of all the NCBI information actually. Okay, and one more thing, whatever the databases I just mentioned before, it's free of cost. All of you please go through it. Okay, next one we'll discuss about how to understand the sequence data. Okay, so as we have discussed about the biological databases mastery is important, you should also know what are the different kind of sequence data format we have. Okay, and how it is beneficial in different kind of analysis, you should understand that first. Okay, yeah, so the one most common sequence format which always we hear is of FASTA. Okay, and next one is FASTQ. Okay, FASTA means nothing but your nucleotide sequence will be there, or protein sequence will be there. Okay, they have the headers greater than symbol followed by gene expression ID. Okay, followed by that, in case of gene or nucleotide base pairs will be there. In case of protein, it has a amino acid sequences. Okay, so if it is a FASTQ, it is nothing but it is very powerful. It has both sequence information as well as quality information is there. This is all about all NGS data mostly will give the raw data in the format of FASTQ files. Okay, yeah. So here the quality scores matters here. You should understand in FASTQ the how the quality scores being given. Based on the Fred scores, the quality scores will be given. Okay, so in most of the analysis, the quality score should be Q30 should be there. Okay, yeah. So if the FASTQ is Fred score is 30 and above, it says that errors will be little less over there. Okay, yeah. One more thing I would like to say, if you take the FASTQ file as such for your research, okay, so that will end up in lot of errors in the result is will end up. We should first check the quality control. Exactly, you should find if any low quality data is there, you should trim up those data. If any false variants will be there, you should trim up that. Okay, and you should take the good quality data to proceed further for the analysis. Okay, yeah, so you should really check The quality control can be very well performed through fast QC tool and multi QC tool. Okay, here we can check the quality control of the sequencer, GC content, or any duplication levels are there, that can also be checked. Okay, yeah. So you just remember that garbage in, garbage out is really important, right? Likewise, good quality data we should take in for further analysis. That keep in mind. Okay, yeah. So one more, just one case study which I wanted to share with all of you. Like you see, without QC, if you perform the result, okay, in 2012 what happened, like the research has been carried out in a lab. Okay, so they have published a breakthrough result stating that they have found out a novel gene variant, okay, which is actually linked to a rare disease. Okay, but after within a month, another group has proved it is just a low quality artifact sequence. Okay, they have not trimmed properly. Okay, so this was in research, if you see, this wasn't just embarrassing, but also the amount they spend in the amount and time which they spent in the research has gone waste, right? So you should use the computational biology tool. Okay, and not just trusting a pipeline, you should also check how it has been evaluated, what kind of results you have got critically. Okay, you should decide on those aspects and you should work on. Okay, yeah. So next important quality any computational biologist should have is mapping a gene to pathways. Okay, yeah. So here we have some tools. Okay, like KEGG metabolic pathways we have, reactome pathways are there. Okay, how it can be used, I'll just tell you. Okay, so once you have processed your data, okay, and you have your gene list or variants in your hand, okay, the real question begins there. Okay, so whether whatever the results or variants I got, is it biologically significant or not? Okay, so here in this case only we should know how biologically they are significant or not, in which pathway it is involved. We should find out that first. Okay, so here pathway analysis came into picture. Okay, so imagine that you have 200 upregulated genes here. Okay, so it is really overwhelming, right? 200 upregulated genes you have. Okay, so but if you map them with the help of this KEGG pathway or reactome pathway databases, you can see 40 of them will fall into one single pathway itself. Okay, so then you can tell which pathways are really involved in those analysis. Okay, so based on this you can move your list, to particular which pathway has more information to that particular disease or that particular biological process. You can clearly tell about that. Okay, so this could be tailored, even if it is involved in any disease mechanism, that can be very clearly dotted out in this way. Okay, so you can think that how impactfully this drug discovery process been happening. Okay, instead of randomly choosing, chasing any particular gene, you can pinpoint that this is having a very good network. Okay, this might have relevant intervention in that disease. Okay, so this is how the computational biologist power will came into picture. If you do the good, pathway analysis will be done. Okay, yeah. So especially if you see in most of the cancer gene discovery, pathway analysis plays a huge role. Okay, especially in the study of EGFR signaling pathway, they have actually driven the tumor growth exactly here. Okay, they have zoomed out exactly the pathway level. They didn't just only confirm the existing knowledge, they also identified some novel genes. Okay, where the same pathway is there, and which is having the potential therapeutic targets. Okay, so here perfectly it tells the computational biologist beyond the genes, it will the pathway really tell whether this mechanism could lead to any kind of targeted therapeutic approaches or not. Such kind of things can be evaluated over here. Okay, yeah. So next one important thing, like nowadays every biologist should have is sequencing. That is NGS data analysis, interpretation, how to analyze that. Okay. So quickly I will let you a walkthrough through typical sequencing workflow, how it will be done. Okay. So I have told the sequencing will generate a raw data FASTQ file will be there. Okay. So the first step is that you should read those. What are the short reads, long reads are there? That raw reads, that is a starting point actually. Okay. The next step which comes is quality control. This quality control can be achieved well through fast QC or multi QC tool. Okay. So here we check the quality of the sequence, duplicates, and also adapters if any remains. Okay. The next step which come into picture is alignment. Okay. So here exactly we can mapping the reads to the reference genome, and then we can also any duplicate marking will be there, that can also be find out over here. Okay. Next step is to identify the variants. Okay. So that can be achieved very well through the uh, gatk tool. Okay. Next, variant calling can also be done through we can also find any specific potential mutants is there or not. Okay. So then variant annotation can be carried out through vep ensemble or ANOVA tool can be done. And finally, if any biological interpretation is there, these variants are related to disease or any kind of therapy it is there, that you can clearly you can choose. Okay. So here what a computational biologist role is that, the workflow is just a mechanical, you should not understand it mechanical. Okay. You should make decision in each step, what kind of results you got, how it is going to align. Okay. How, which filter we need to do, which database version I should use. Okay. Each choice of yours will impact your downstream analysis result. Okay. That's why employees, value the people who not only run pipeline, who actually understand the real aims and objectives of the work, okay, behind each step. Okay. So in most of the cancer genomics research, they especially use workflow to find the driver mutation. Okay. If you see in the COVID-19 sequencing has been done, okay, over the pandemic's time, it has been done. Okay. So here, you know, do you think this mechanical help of data analysis has helped here? No. It is, the real world pandemic is going on, and the person who can understand the virology and also the computational skills, tracking the pandemic, that have made the situation handled very well in vaccine development, in many other things, and identifying the mechanisms. Okay, tracking those mechanisms. Okay, so without this, it wouldn't be possible. So you should be a right computational biologist in doing all these tasks. Okay. Yeah. So now we are coming into important thing. Everyone should have a note on this. What is the career essential skill set checklist you should have? Okay. Now let's talk about careers. What skills should put in your tool list or toolkit? Okay. Yeah. First thing is database literacy is more important. Okay. So you should know how to query, integrate, or interpret multiple data resources like NCBI, Uniprot, Ensemble, etc. You should know. You should also know some specified databases also. Okay. Next thing you should aware of sequence analysis. Okay. So in sequence analysis, you should know how to do the quality control, alignment, how to perform, identify the variant, how to, okay, and these all things you should be thorough. How it is associated with the disease, you should have an idea on those things. Okay. Yeah. So next come is your structural bioinformatics plays a huge role. You should have a, how a protein modeling is done, how the protein structure prediction will be done if three-dimensional structure is not there, how to perform the molecular docking studies. Okay. So how the molecular interactions being done. Okay. You should be thorough enough of all these concepts. Okay. Next, data visualization, how to do, especially, significant large result you have in your hand. How beautifully you can show it as a volcano plots or networks, heat maps using the Python, R programming, and Cytoscript software. How efficiently you can show all these things. Okay. Yeah. When you are visualizing your data, it give a better clarity for the data. Okay. And also you're making your co-learners and also other researchers, you're convincing them and you're giving the path to fellow researchers as well. Okay. Yeah. So next thing which comes into your picture is that reproducibility, whatever the pipelines you have made, that should give a, you should make a good GitHub version, notebooks. Okay. You should maintain those all things. Okay. Your, whatever the pipeline you have developed, it should be reproducible one. Okay. So these, whatever tips I'm saying, it is not an optional anymore. Okay. Everybody, even your wet lab life science researcher who is listening in front of me, definitely you should adhere these skills in your, checklist. Nowadays, as I said, people are really looking for a hybrid scientist and researchers everywhere. Okay. So if you want to be employable, please develop these skills in your own way. Okay. Yeah. So now how to build a career ready portfolio. Okay. Let's have a quick look on that. So, how you can turn down your skills into opportunity means you should have a very good portfolio you should maintain. Okay, so how the portfolio will come? Is it like a picture portfolio? Yeah, something like that. Okay, so how in computational biologists can make a career ready portfolio? Let's see that. Okay, so employers, they don't just want to hear that I know R, Python, docking software, I know this protein software, etc. Nowadays. Okay, they want to see what you have projects or anything you have successfully done by using your computational skills or programming knowledges, how you have effectively used it and what are the good outcomes you got out of your projects, that they wanted to hear. Okay, now let's take some examples, like few mini projects you can do, like small RNA sequence project, where you can show identically any differential express genes are there from the public data set you can take, and you can show some variation over there in the pipeline development. Okay, you can do small work like protein protein interaction network for a disease gene using any kind of software you can make it like cytoscape, etc. Okay, you can also develop an interactive dashboard like plotly and shiny to show the QC results, data visualization you can show. Okay, so whatever you have done, you should document very effectively. Okay, so whatever the pipeline you have, even small thing you have done, please document it in a GitHub. Okay, and also in GitHub you should include a README file which can explain what the work you have done, what is the biological question you have taken, which data source you have taken, what are the steps being used and interpretation, etc. Okay, so now your portfolio will turn your skills which is invisible into a visible one. Okay, yeah, so to become a computational biologist, okay, so it is not just only mastering every tool. I'm keep telling that, keep this in mind. It's about adapting a mindset. Okay, so you should start with a biological question every time. You should choose the right computational approach. Okay, keep learning in small but with the consistent steady steps you keep learning. Okay, share your work openly to everyone in transparent way. Okay, and most importantly that keep practicing. Okay, don't wait until you feel ready. Okay, take a data set today. Okay, run the QC of it and then post your results. Okay, keep iterating these steps. Okay, yeah, so I would say that biology, has gone computational actually. Nowadays, every employers need a hybrid scientist. Okay, so you should have a right database sense. Okay, sequence literacy should be there, and pathways are your foundation. Okay, so keep this in mind. Use the right tool, choose the right object. Okay, build your good portfolio to showcase your journey. Okay, so my challenge for all of you today is that you should start one mini project this week. Pick any data set of your interest, run small analysis, anything you can, and share it. Okay, this is how you can move beyond the pipelines. Okay, thank you so much everyone for listening patiently. Okay.
Thank you so much, ma'am, for this informative session. There was a lot to take in actually, and I like that you gave them a challenge, saying that like work on some mini project this week. So it's a motivation for them to get started. So I hope all of y'all keep that in mind. Ma'am has also told y'all what are the skill sets you'll need, and also how you should start your own, work on in GitHub. So all of this and more, y'all can check out. I hope you all know that we have a bioinformatics internship that is starting soon. So all of this and more will be taught to you over there. So if you're curious, if you're confused, or you're wondering how do I get into this, so do check it out. The link will be in the comments. So thank you so much, ma'am. Now next up we have our next speaker, Dr. Neeraj. So it's all over to Dr. Neeraj right
So let me just share my screen. I hope I'm audible.
now. Yes, sir, you are.
Is my screen visible?
Yes,
Could
yes.
you? Yes. So thank you for the opportunity, and I think it was a wonderful session by Dr. Elamathi. She has given a very deep insights regarding bioinformatics, and she has majorly focused over various tools, various databases, as well as strategies to follow in bioinformatics. And she has also discussed about computational biology. So today, I am, as I have already been mentioned, Dr. Neeraj. So I am going to talk about the bioinformatics toolbox. So the skills or the tools which you are familiar with or you have hands-on experience with, you can work well with, so that that can bring you the job, and that can help you to get hired in industry or in academia when wherever you are seeking a particular job as per your interests. Okay, so we will have a brief overview and importance of learning these tools, right? So, why bioinformatics tools matter? Okay, so bioinformatics has become central role to life of life sciences, pharma and healthcare. Okay, so employers in academia, biotech or pharma are not are increasingly looking for candidates who can not only work with biology but uh, can also understand and have practical experiences with bioinformatics tools. Okay, so having an hands-on experience that shows that you can analyze, you can interpret results, and you can contribute to the modern research and innovation. Okay, so bioinformatics is no longer just a field; it is the core field. Okay, that have been evolved over a period of time, and I think Dr. Elamathi have already been mentioned the importance of bioinformatics and understanding of bioinformatics, right? So employers basically seek candidate with hands-on tool experience. Okay, so how bioinformatics is accelerating various domains in life sciences. Okay, so it is accelerating drug discovery. So bioinformatics can help in identifying promising drug targets, model molecules, and can predict interactions much faster than the traditional lab-based methods, right? And then it is also accelerating the genomics and proteomics, and regarding genomics and proteomics, Dr. Ilamathi have elaborated in depth already. Okay, and if we come to precision medicine, then bioinformatics tools as well as approaches or strategies can analyze patient specific genetic and molecular data, where bioinformatics tools can support personalized treatment strategies that can be more effective as well as have fewer side effects for that particular patient, right? So, what is the career value of bioinformatics? So, learning tools is equal to employability and research readiness. So, if you have an experience of bioinformatics tools, an expertise regarding any bioinformatics approaches, whether they are structural bioinformatic techniques, whether they are multi-omics techniques or just genomic techniques, then it will create a wide horizon, and it will open various doors for you in academia as well as in research domain. Okay, so that is how the bioinformatics tools are important and why you need to learn these tools. So what are the, so I am going to focus over structural approaches mainly, so structural bioinformatics techniques, right? So homology modeling. So as Dr. Ilamathi said that if you have a particular protein sequence, then how to predict their structure. So while going for drug discovery technologies or understanding protein dynamics, or you wanted to understand the nature of specific protein, or you wanted to characterize a particular protein, then you need to have a 3D structure, right? So these days we have the various technologies, okay, to predict the hypothetical structure of protein. For example, the alpha fold, rosetta fold, you may have heard of these things, right? But the core technology or the core approach which works underneath of for these alpha fold or rosetta fold is the homology modeling. And being a structural bioinformatician, you must have understanding of homology modeling. So, what is homology modeling? So, homology modeling is the prediction of 3D structure from the related templates. Okay. So, here it is a method which is used to predict the 3D structure of protein if the protein structure is unknown. So, if a related protein with a known structure, which is called the template, we just try to find these templates by various techniques like protein blast and other approaches. So we can just try to build the 3D structure of our protein by considering that template. right? So this technique is called homology modeling. Okay, so this is the process by which homology modeling works. So we have the query sequence, the sequence you wanted to build, for which you wanted to build the 3D structure, and you have tried to find out the template, which is the homology sequence. So if this sequence should be identical or similar to the query sequence, and then we apply computational techniques, and then we have the predicted 3D structure for our protein. So that is how the homology modeling works. Then we have molecular dynamics technique. Okay, so molecular dynamics simulates the atomistic motion over times that assess the stability, interaction, and conformational changes. Okay, so from name itself, you can understand that what is molecular dynamics. So whenever you are trying to study a molecule in a dynamic motion, or because in homology modeling or in molecular docking, you basically studies the static motion of a particular molecule. However, in molecular dynamics, we completely focus over the a particular molecule, whether it is a protein molecule or it is a complex with the ligand, then it always stays in the motion for the time period which have been mentioned, or the MD simulation has been carried out, right? So, MD simulation, where the proteins are not static, they move and change their shape in real biological conditions. So, MD simulation use physics-based models to replicate the motion of atoms and molecules over time. Okay, so it allows the scientists or researchers or academicians to study protein flexibility, stability, interactions with ligands, and conformational changes. So this is also a core approach for structural biology or structural bioinformatics. Then we have the MD. So MD directly supports the structure-based drug designing. So whenever we wanted to understand the stability of a particular protein, while if a particular ligand is being interacted with a protein target. Okay, so how it is interfering with that particular ligand is interfering with the dynamic motion or the stability of a particular protein, right? And the second thing, how a particular ligand is interacting with protein in a dynamic phase, because in human body the whole body, the molecules at atomistic level always stays in motion, right? So if we wanted to understand, we need to simulate the human system in computational setup, that is called the molecular dynamic simulation, right? And that is how it supports what you say, the drug discovery technologies. Okay, then, what are the tools for computational structure prediction? So, as we have discussed about homology modeling, so that is the underneath principle on which the structure prediction techniques work. So, we have various tools for to carry out the structure prediction of any protein sequence, right? So let us just first go through the flow, that how these tools work. I have also already explained it in brief in previous slide, but let us just jump in there again. So first, we have a query sequence. So query sequence would be your protein sequence you wanted to build model. So build model the 3D structure, right? So this is your query sequence. So now for this query sequence, you wanted to search, or you need to search the template structure first. So it is the identification of a related protein with the known structure works as a beginning point or initial point in homology modeling or the structure prediction. Then after having a suitable template, you need to build the model, and then you need to carry out the assessment. Okay, so in this step, we or the particular tools tries to generate a 3D model of target protein and evaluates its quality, right? And whenever we have the protein structure, we carry out the molecular docking, right? So here we predict the small molecules, a particular ligand or drug like molecule binds to the protein's active site. So the purpose of carrying out the molecular docking is to find out that where exactly the binding site lies, right? And we can also carry out the MD simulation for post stability and refinement. So in initial phases we carry out MD simulation just to minimize the protein energy. So the build structure may have the higher energies that can create an instability in the protein structure, the predicted protein structure. However, the MD simulation can be carried out, which can refine the structure as well as provide more stability by minimizing the energy. And further, we can also implement MD simulation to assess the molecular docking interaction studies, right? So we have various tools that can be carried out for that can carry out MD the homology modeling. So first we have Swiss Modeler. So it is an automated server, and it is available online. You can just Google it out, and it would be available for your assessment. Okay. Then we have Modeler. So it is a tool which can be operated manually by the user, and you need to have the basic understanding of Python programming while working with Modeler. So it is a comparative modeling by spatial restraints, right? And then we have so these are the symbols for Swiss model, and this is the for AlphaFold. So AlphaFold, we also have AlphaFold, which is a deep learning based prediction. So it is also an online web server, and which basically works on the principle of deep learning. Okay. And which have recently comes in spotlight. Okay. So and it is AlphaFold is trying to predict the many of the structures which have not been resolved earlier by X-ray or NMR crystallography. Okay. And it was a breakthrough in structural biology techniques, right? So these are the homology modeling tools. And we can also work with the molecular dynamics tools. So we have GROMACS. So GROMACS can carry out the high performance MD simulation studies and analysis. So the GROMACS can be implemented here in this step. And for your information, so GROMACS is an open source tool, and it is available freely. You guys can work out and can start working with the GROMACS if you wanted to understand the MD simulation techniques, right? So we have Amber and NAMD also. So they are also the molecular dynamics tools, but if they are mostly integrated as a force field in GROMACS. If you wanted to work with Amber or NAMD independently, then you need to carry out few registrations before working, right? So basically Amber and NAMD work as force field primarily in GROMACS, and they are already integrated in GROMACS, and this is the symbol of GROMACS, right? So I hope you these things are pretty awesome to know you. And if you are getting learning, then you need to start working with all these things. And these the most of the things are available online, and as a web servers and web resources, so you can start just browsing them, like just Flipkart or Amazon. If you wanted to shop something from Flipkart, you can also you can also go to web source or Google Chrome or any Firefox. Just try to type out the Swiss modeler. Okay. And try to look out for it, that how it is working, how a protein sequence can be directly converted into a 3D structure. So that would what you say, force your or push your curiosity regarding understanding of such techniques, right? So we have various tools which are directly focusing on drug discovery. Okay. So before jumping directly into drug discovery, I wanted to tell you something about virtual screening and docking. So what is docking? Okay, so docking is whenever you have a protein structure. I think Dr. Elamathi was also talking about docking. So whenever you have a protein sequence, so it has been converted into 3D structure. If not available, then you have a 3D structure of a protein, right? So why we always talk about protein? Because at a functional level, whenever there is a mutation or whenever there is any changes in genomic at a genomic level, then they are directly translated into proteins, they which comes in action for the functional part of a human body. Okay. So I think you are understanding that why proteins are necessary and how they are, why they are more significant in understanding, or they are the drivers in drug discovery. Okay. So whenever you are talking about a particular disease, then you try to find out that what are the proteins which are participating in the progression of disease, or which are leading to that disease. Okay, so whenever you have those faulty proteins, then you try to find out their binding site, and you try to dock particular ligands or drug-like molecules which can interact with the binding pocket of that protein, and can carry out their biological effect. So those biological effects could be the stimulation of that particular protein structure, or the inhibition of that particular protein structure, right? So to carry out or to understand the interaction patterns between a protein and a ligand, the technique which is used is called docking. I think it is clear. And so docking can be carried out for 10 or 20 molecules manually. So whenever you wanted to find out the lead molecules or a potential drug like molecules from thousands, or from 10,000s, or from 1 lakh compounds, or 10 lakh compounds, then you cannot carry out that approach manually. Okay. 10 20 can be carried out manually. You can just change the script, and you can just carry out the docking process. However, whenever there are 10 lakh compounds, then you need to fix this process, and you need to automate this process. So the automation of that process is called virtual screening. So you are screening a library of a number of compounds and number of compounds uh, just by virtually screening them. Okay, because you are not carrying out any wet lab experiments for screening those compounds, that what are the potential lead compounds or not. So that is called virtual screening. And for a docking, we use Autodock. So Autodock is a tool which is used for docking studies. Okay, so it is a popular tool, open source tool, and widely cited in research. And most of the docking is being carried out in Autodock and Autodock VNAP. And these days the diff doc, which is a deep learning driven tool on the diffusion models, right? Then if you wanted to carry out the virtual screening campaign, then you need to use the Pyrex. So Pyrex is a user friendly interface, have a user friendly interface, and it can integrate docking with the databases. So it can also integrate the various chemical databases like Campbell, the PubChem, etc. So you can also fetch out the browse the Campbell, or the CHEMBL is the Campbell, and the PubChem, which are the major sources of chemical compounds. And there you can see that how vast is the chemical space, the number of compounds that could be the potential lead compounds or the potential drug compounds. So the potential drug compound is a compound that can be transformed into drug, that is not exactly a drug, that need further optimization. So these things should be clear to you before working. So this is the interface of Autodoc. Okay, so this is the protein structure, and here you just optimize and you just visualize the binding sites, and then you create a grid maps here, and then you need a further those grid maps would be further needed for defining the grid boxes for ligand binding. So the applications of docking and virtual screening is for screening compound libraries by using virtual screening and predicting binding affinities. So how strongly a particular ligand or a potential drug-like compound is binding with that particular protein structure. That is the application of these tools and these technologies. Okay. So, what is the career perspective in docking as well as virtual scanning? So, why they are significant to learn these techniques? So, what is the significance? So, significance is, so skills in docking can accelerate entry into pharma and biotech roles. So, if you are working in silico drug discovery and you wanted to just enhance your or you wanted to enhance your skill set and wanted to absorb in this the pharma tech or the biotech industry, then you must have such skills, the docking and virtual scaling. And the next skill is also the visualization and analysis. So whenever you have docked your compounds, you have carried out the virtual screening campaign, then you also wanted to analyze that how particularly those compounds are getting interacted with the binding site. For these you again need tools. So the tool you have is the pymol, so which is widely used tools a tool for understanding, for visualization, for just to manipulate your structures, to understand that how a particular ligand is interacting in the binding site, what are the surfaces of that particular protein. Okay, what are the potential gradients of a particular protein, whether it is falling in a positive range or a negative range. Okay, and what are the steric clashes. Okay, and if you wanted to visualize that protein structure at stick models or ribbon model or cartoon level, so all the integration or all the facilities would be provided in pymol. So it is a 3D visualization of protein ligand complexes, and it also produce essential for produce publication quality images and presentations. So this is the interface of pymol. So pymol looks like something like this, and it has this is the viewer window where you can just watch out your protein. So if this image could not be zoomed out more, however, you can see, sorry, you can see here that this is the ligand which is shown in a sticks model, and this is the cartoon representation of a particular protein. So that is how, and this is how you can just integrate pymol in your studies, right? And the other tool you must have you must learn, you must have skills with, is cytoscape. So cytoscape is used to visualize the biological networks and pathways. Okay, it can integrate multiomic data with molecular interactions. So it can integrate multiomics data like genomics, proteomics, metabolomics with molecular interaction networks. So it provides, or it facilitates as a central tool for systems biology and pathway analysis. So if when you wanted to narrow down the number of genes participating in progression of disease, what are the faulty genes, or what are the significance of genes, and how they are interacting with other genes, you wanted to understand the network biology of a particular genomic interactions across proteomics, genomics, and metabolomics, then you can use cytoscape. So significance of learning the bimol and cytoscape. So both these tools are highly cited. Okay, because they are they plays a central role in structural biology and the functional bioinformatics. Okay, and they often required in job posting. So whenever you are looking out for the bioinformatics jobs, then you would find out that the need, or they required a person with the integration who can bring up the skill set in pymol or cytoscape. So how you can choose your niche in bioinformatics? Okay, so you can map your interest areas. For example, if you are interested in genomics, you have interest in genes, sorry, in DNAs or in RNAs. So you can start working with NGS pipelines. The tool is Galaxy, which is used to visualize the genomics or the genomic networks, and Bioconductor, which is an R package, which will enhance your skill set in genomics and would help you in understanding the genomics well. Then you have proteomics. Okay, so tools such as MaxQuant and this Perseus help in analyzing protein expression, modifications, and quantitative proteomics data. So these tools can be implemented, and you can learn that how to work with proteomics data. Then in structural biology, if you wanted to work in structural bioinformatics or structural biology, then Pymol, Romax, Modeler, Autodoc, Vina, etc. are the tools that you need to learn, and you must have hands-on experience, and you can just be an expert in these tools. Okay, So, while working separately in all these domains, by like genomics, proteomics, structural biology, you can have the stack of these skill sets. So you can combine one or two niche tools, and you can use generalist tools. For example, the Pymol and Cytoscape. While working in structural biology and proteomics, you can also learn Pymol and Cytoscape simultaneously, and you can also develop in-depth knowledge in one area and breadth across others. So have an be an expert of one domain, and you can have the basic, or what you say, basic to advanced knowledge of other domains, which would help you to grab more opportunities, as well as with your skill set you can work at the cross disciplinary areas, right? So, what basically employers look for? So, specialization in one domain and versatility. So, if you have one or two domains at a time, you can work at genomics as well as structural bioinformatics. Then you are a good fit, or you would be the best fit for for a particular employer. So we have a few career tips while you wanted to look out for opportunities in bioinformatics. So have hands-on skill as compared to theoretical knowledge. You must have theoretical knowledge, but the hands-on skill set helps you to get hired easily, right? The second thing is start with free and open source toolkits, then specialize. So there are widely used tools like Pymol, Cytoscape, Galaxy Bioconductors, which are freely available. Okay, so you can start working with these tools from today, and you can have, or you can gain confidence. And then by gaining confidence, you can keep working on these tools, and you will have specialization while working on these tools. Then you can also while working with the tools and having specialization, you can show your tool proficiency in your CV or in your LinkedIn profile. So how would you do that? You can create mini projects, as Dr. Elamathi just suggested you and given you a task for developing a small project at your own. So you can develop those small projects or mini projects at your own, and you can start working with these tools. And whenever you have outcome of those tools, you can showcase those characteristics or skill sets in your CV, in your LinkedIn profile, as well as in your GitHub repo. Okay, so you can just show off all these tools, skill sets in your interviews being carried out by any employer, which would help you in getting hired and can enhance or increase your chances of hiring. So that is all from my side, and I wish you all the very best while going through and learning through these tools. So thank you so much for this wonderful session, and over to you, Dilakshi ma'am.
Thank you so much, sir. So I hope this was very insightful for all of y'all. So quickly post your questions if you have any in the comments below, because we're a little short on time right now. So we'll just take some questions. But this was such a great session because sir and ma'am, both of them have given you so many insights, like, what are the checklists that you need to have, the skill sets, and even the career tips that sir has shared with you, saying that, you need to, now your hands-on technique is what is more important rather than your theory. Now that time has gone for theory, hands-on is very important. And how there are so many open source or free software also which is available for you so that you can experiment, like how sir said, like how you all browse on Flipkart and Amazon, you can even, spend time over there. So this is how, you build that interest in bioinformatics. It's now or never, you can't run away from it now. And then also coming to making mini projects, like ma'am has already given you a challenge, so why don't you all work on it? It's something new, right? So you all are beginners, you all are showing that enthusiasm, you all want to become job ready, career ready. So this is a time for you. So these sessions, attending these sessions will help you because you get certain tips like this. Now, if you want to be guided and you want to be mentored and you want to go more deep into it, you want to know more and not just, fumble around, then make sure you check out the Bioinformatics Research Internship, which we have, which is starting soon. That is the 30th of September. So the links will be posted in the comments. So over there, you'll get your hands-on training, all those tools. You'll also be working on projects. You can choose like 3 months, 6 months, 1 year. You'll be getting, work experience because of that. So all of these mentors in front of you that you can see on your screen will all be there to guide you. So if you want to know more, do check out all the links in the comments. So if you have any questions, please let us know. Put it in the comments. We'll answer a couple right now. So just a couple that I have found is so according to both of y'all, or any one of y'all can answer, which bioinformatics tools or database you feel is most underrated but is highly useful for beginners? Any insight? Do you feel anything is underrated? Dr. Ilamathi or Dr. Neeraj can answer.
According to me, as nothing is underrated, but still I would say Galaxy server for NGS data analysis. Most of the people, it is easily doable, but most of them are not using it. If they can use it, that will be very good. As well as some server, browsers like UCSC browser, as well as IGV visualization tools. It has not been most commonly, it is not used by the peoples actually. So like some of the visualization tools like Rasmal, Chimera, they have their own benefits, but it is also not used that much. It can be used according to me from my end. Yeah.
Yeah, so see, you get all of these things. So if you'll join the internship, y'all will learn more, and you'll be working on all of these tools, and you'll get a better, expansive view of what and all is being used, and how sir said, you should be versatile. Specialization plus versatility, that is what everyone demands. So, ma'am, do you think small projects are enough to demonstrate capabilities, or do recruiters expect, larger, much bigger, this, expectation is more? People should have worked more.
Um, I would give you an example. If you done even a small project, with better clarity of your aims and objectives, with the right choice of using the tool, if you can able to interpret, that makes sense. Instead of making too much pipeline, too much tools, too much big data set, too much objectives without any clarity in interpreting your result, that doesn't make sense. I have seen my own students, they go for the interview, they did developed a good variant calling pipeline and all those things, but sometimes they fumble in the interview. They ask what are the different types of variants you have, how do you identify, how do you interpret, how you clinically correlate. They could not tell such small things, but they developed so many things, okay, without paying attention towards such things. So doing a small thing, that makes sense, like how we in the cooking, how we use mustard, that even gives turmeric, small things which we add, that makes much sense, instead of making very big thing. So I would say, like, understanding of the things with the right choice of tools and using the small things makes much sense. And how to correlate with the biological significance, that matters a lot. Yeah.
Because many people may be scared or apprehensive, or like, oh, okay, I don't have much, is this enough? So that's why this question came up. So see, make your foundation, your basics clear. And like ma'am said, with something small, that is enough to make a huge impact. So once you have all that clear and settled, then you can definitely answer any interview question confidently. So don't hesitate thinking that you're too late, or you're lagging behind. Make a start. Okay, so make a start this week. Now, one last question, ma'am, do you have any, feeling about which sectors do you think you'll see the most demand for computational biologists, like pharma, agriculture, healthcare, anything you feel,
So now the demand is in pharma, as well as if you see all of the omics sector, I would say like genomics, proteomics, okay, data analysis, data scientist, okay, huge data is on the line. It's on the credit. So in healthcare sector, people are looking for, because precision medicine on its way to India and everywhere in the card. So people are looking for data scientist, and as well as pharma is also doing some of the therapeutic approaches at more at its end. So these are the sectors where now most demand is there actually. Yeah.
Yeah. So thank you so much, ma'am. So now let's end this session. But before you leave, we have our free career guide for you. So you're a beginner, you're struggling to figure out what this bioinformatics is all about. You want to read some papers and all. So check that guide out. The link will be posted in the comments, so do check it out. Do get your guide. So thank you so much for attending this session, and don't forget to check out the bioinformatics research internship that we have. All of these mentors that you saw on screen will be there to guide you, to help you. You'll be working on projects as well, so you will get that experience and that confidence and get career ready in bioinformatics. Because remember, the future of biology is computational, and it belongs to those who start building their skill sets now. So if you are one among them, check out all the links in the comments, and we will see you next time. Okay, thank you so much, ma'am. Thank you so much, sir. And thank you all of you for joining us. See you in the next session, guys. Have a nice night.